Our next speaker is Dr. Kameshwar Rao Badri. He is going to talk on past, present, and future of therapeutics of obesity, a long ignored chronic disease. He is currently working at Morehouse School of Medicine, Atlanta, Georgia. Dr. Badri, floor is yours.
Good evening. I thank the organizers for giving me this opportunity, and I thank you all the audience for staying so long listening to all of us who are placed at the end. So, title of my talk is Past, Present and Future of Therapeutics of Obesity, a long ignored chronic disease. And of course, what I will be focusing also on targeting cell signaling mechanisms and use of natural compounds in adipocyte plasticity in the metabolic evolution. So all of you know, obesity is a chronic metabolic disease. Of course, as I mentioned, long ignored, meaning that it is because of overeating or excessive positive energy. So it is characterized by excessive adiposity as measured in BMI, kilograms of weight in meter square. So it is a comorbid condition for many of the diseases. So if you look at this BMI, if it is less than 18.9, so it will be underweight. 18.9 to 24.9 is a normal weight. 24.9 to 29.9 is overweight, and 30 to 34.9 is obesity, and over that is chronic obesity or extreme obesity. However, this is a universal scale, but when it comes to Asians, there are certain bodies suggested 23 to 24.9 will be considered as overweight, and anything above BMI 25% will be considered obese. And all of you know, obesity is associated with many of the physical related diseases like inherited coronary heart diseases, type 2 diabetes, stroke, etc., etc. Not only that, it is also associated with emotional symptoms. So it leads to even a lot of stigma in the people, anxiety, and different things. So when you look at like obesity, it is becoming already it's a global pandemic, and as you see here, US statistics, self-reported obesity shows that from 2011 to 2018, so the incidence of obesity percentages are increasing. Almost the red color, dark red color that you see there is 35% of the population in those states are obese, and now it is even more horrible. So overall, right now in the US, 2/3 of the population is either overweight or obese, and if you look at like obesity rates, almost 40 to 42% of population is obese. So just to go over, look at the certain period of time since 1975, so the obesity rates have tripled, and overweight men, women, statistics remain pretty much standard. However, if you look at obesity rates of women and men, it's like a constantly rising. More alarming is this is also happening in children and adolescents. That is like really very alarming. And even if you look at like obesity at a global level, it's like kept rising, and now we have to like seriously focus on this issue. And when it comes to etiology, so of course majority of times it's nothing but like excessive positive energy balance. So it could be various reasons for overeating, low energy expenditures because of all these different reasons, factors mentioned here. Physical inactivity. Not only that, there are like various biological factors as well as environmental factors. They are going to like contribute for this obesity. So it's no more just energy balance, but this situation in multiple ways. So when it comes to obesity treatments, so if the BMI is a little around 25-ish, so it is very good to start focusing on physical activity and behavioral therapy, healthy eating habits like lifestyle modifications. And if the BMI starts going up like about 27, it's better to start pharmacological management. However, we are going to look at some of the pharmacological managements that are available, which is not very promising. And anything higher than 35 BMI, which is like a chronic obesity and associated with other metabolic complications like diabetes or cardiovascular issues like hypertension, that's like where bariatric surgery is advised. So if you look at the history of these different therapies, pharmacotherapies, so since 1900s there were like different therapies available. So starting with thyroid hormone, so crude extracts of thyroid hormone used to be used in those days, but now it makes sense because thyroid hormone has like these T3 levels, and now there are new mechanisms identified how these T3 is going to regulate body weight. So as you see, different drugs were identified over a period of time, and if you pay attention, many of them were withdrawn. So it is mainly because of the side effects associated. I'm not going to go into detail on all these drugs. So if you look at liraglutide, which is like a GLP-1 agonist, so technically this is being introduced to treat the diabetes, but not like the rest of the diabetic treatments. This one, in fact, is reducing the weight. So that is one of the reasons why this is being also used as a in the management of obesity. So if you look at these different drugs, orlistat is the only one that will be like working at the digestion level, reducing the activity of like it inhibits the lipase activity, which is involved in digestion of fats in the intestine. So thereby, like these patients generally they are going to defecate majority of the fats that they eat along with this drug. However, the side effects include like steatorrhea, leaky, oily stools, and all these things, flatulence, and there are like so many side effects associated. And if you look at the other drugs that are mentioned here, in one way or other, so they are associated with acting on the brain. So either they will be like acting on the hunger centers of the brain or satiety centers of the brain, or they might be acting on serotonin, acting as a serotonin agonist, or they are stimulators of synaptic noradrenaline, dopamine, and serotonin releasing activity of an nerve brain. So they also will be like acting on opioid receptors, and all these like pretty much it is easy to say it is messing up with the brain. A lot of people think that way, and including I'm one of them. So technically each one of these they have like a moderate function. So you will see like a reasonable amount of weight loss, but it's not very significant. At the same time, they are associated with these different side effects. And of course, bariatric surgeries, there are like different types of these bariatric surgeries are available, laparoscopic, additional binding, and sleeve gastrectomy, where you can see the cutting off of the like a major portion of the stomach in panel B, and also you can see like Roux-en-Y gastric bypass surgery, where like the first portion of the stomach in portion C, like panel C, you can see the front portion of the stomach is connected directly after the duodenum, where like majority of the food will be like without digestion will be sent to the latter portion of the small intestine, so caloric intake will be relatively less. So similarly, there are other types also. But it is well proven that in all these bariatric surgeries, so one has to maintain strict dietary restriction, and they also have to have this healthy lifestyle of habits. So that is one of the reasons why many of these people who are undergoing these surgeries generally they will be like regaining weights later in time. Of course, there are tons, as one of the presentations this morning was mentioning, like it is, I think $60 billion market or something like that. So there are like so many supplements, but some of these supplements are being used in India too. So one of them is Garcinia cambogia. But there are like tons of literature showing that this is like a hepatotoxin, but still we are using. So when it comes to like some of these supplements, definitely one has to like make sure, study them thoroughly, systematically to make sure the safety of these supplements before they are like officially accepted by the local people. So when it comes to adipose tissue, of course for a very long time we know only fat is bad. So because we know only one type of fat exists in humans, at least at a certain point of time recently. But lately there are like new types of fats. So that is like white fat, which is bad. So white fat is like basically whole body energy. It is involved in whole body energy homeostasis, energy storage, fatty acid release, and it also has a endocrine function. So it has other like simple protection mechanisms for the internal organs. However, there are like other types of fats are identified. Brown fat is one of them, which is also considered as baby fat, which is present in human newborns, and hibernating mammals have high levels. So its main function is thermoregulation. So it will be spending energy. Brown fat will be spending energy, while white fat will be storing energy. So, of course, when you look at white fat depots, white adipose tissue depots, these depots also have another type of fat cells known as beige adipocytes. So these are like between white fat and brown fat. The name for brown fat is because it is brown, because the presence of enormous number of mitochondria. So because of the presence of mitochondria, they will be like utilizing the energy. So if you look at the adipogenic pathway, so all these cells, white adipocytes, beige adipocytes, and brown adipocytes, they have like a common mesenchymal stem cell precursors, and based on like a different transcriptional factors, they will be like either going to develop into white fat or beige fat or brown fat or skeletal muscle. So during development also like up to certain level, so the development of fat will be like a multiplication of these cells from precursor cells. And when in any stage, if there is like excess energy intake by the organism or a human being, so these cells are going to become hypertrophic. So they are going to like become very, very large. So hypertrophic adipocytes, they lose their function. They become dysfunctional. That is going to cause like an insulin resistance and diabetes. So adipose tissue has multiple functions, and beige and brown adipose tissue. So all of you know about TCA cycle. In TCA cycle, high energy molecules, NADPH plus H plus and FADH2, they will transfer their electrons, and electrons will be transferred from these different electron transport carrier proteins. And at the end, the electrons will be taken by the oxygen, and the energy will be utilized to drive these protons from inside of the mitochondrial matrix to outside of the inner mitochondrial matrix. So while these protons come back, so these protons are going to be like more helpful in synthesizing ATP, because they are going to be like come through ATP synthase. However, in brown and beige fats, it is going to come back through UCP1, which is instead of synthesizing ATP, energy will be like leaked in the form of heat. So there are like different places where this obesity mechanisms, associated mechanisms can be targeted. The digestion level, or fat cell development level, or energy expenditure level. So medicinal plants have like a very successful story. There are like multiple drugs have been isolated from different plant products, and of course today's highlight is curcumin. And I've been involved in like a drug discovery. I've been like isolating different. I have isolated like a different active principle from various plants, and we published like many of these things. And so there are like multiple. So now the new new strategy is to how to convert this white fat or increase the beige fat composition phenotype in white fat itself. So when you look at white fat, there will be like a white adipocytes and beige adipocytes. So if the beige adipocytes are more, that person is going to be metabolically healthy. If beige adipocyte numbers go down and white adipocytes are more, they are going to be like metabolically unhealthy. So there are like multiple plant compounds are shown to act in different levels. So I kind of like focusing on trying to develop drugs that can like affect the adipogenicity or convert these white adipocytes into beige adipocytes. And I'm also working on a protein known as P311, which is a small protein, and this doesn't have any known motives except like a pest motive which targets this proteasome degradation pathway. Initially it was shown in smooth muscle and brain, but I have shown this protein present in arterial system, and these knockout mice, universal knockout mice, they are hypertensive, and I ended up showing that this hypertension is because of vascular reactivity issues, and there are like about 20 figures. I'm not going to go over them, and we published this one and JCI in 2013. While I was conducting that study, what I observed is knockout mice had the increased fat, body fat. So with that, I started looking into like a role of P311, and right now recently we published that P311 is involved in adipocyte development. So here you can see cell culture model. We treat these cells with adipogenic cocktail, they will become fat. Within 5 days, you see increase in P311. Of course, the other master regulators like PPR gamma, master regulator of adipogenesis, is also increasing, and P311 expression is increasing like continuously as the cells differentiate into fat cells. Knockdown inhibited adipogenesis, and then for the first time we showed, before everybody showed that P311 is only cytoplasmic, but in our studies we showed that it is present in nucleus also. It just concentrates to the nucleus before the cells start differentiating. So with that background, like I started looking into murine fat tissues. So P311 is present in subcutaneous fat, gonadal fat, brown fat, and perivascular fat. And to my surprise, when I checked the weights of these fats, there is absolutely no difference in gonadal fat weight and subcutaneous fat weight between wild type and knockouts. That was surprising. I didn't expect that. However, brown fat is like a little lower in knockouts. So I started looking into like why this is happening, and I looked at the cell number. So when I looked at the cell number, P311 knockouts has a less number of adipocytes. So think this way, weight is same, but cells are less in knockout, means their size is larger. So I started counting the volume of these cells, and so P311 knockouts have hypertrophic, what you can call, adipocytes. So next I asked, what is happening with the UCP1? Now I understand. Quantity of the fat is same. Is there any change in the quality? So when I looked at the quality, so UCP1 is a marker for beige or brown adipogenesis. So P311 knockouts showed in brown adipose tissue, perivascular adipose tissue, and also in subcutaneous adipose tissue, reduced UCP1. Of course, we are not going to focus on BAT and PBAT, but the subcutaneous white adipose tissue, the browning is down in knockouts. That means beige adipogenesis is going down. That means quality of the fat is going down. So that concept I verified for the glucose, and these knockout mice are hyperglycemic, and they are showing like intolerant glucose intolerance, as you see here. So pretty much. And I got the doubt, so what is that?
May I request you to kindly summarize?
Yes, sir. Done. This is like 1 minute. So next, so only thing why I asked is why I'm not seeing fat differences, and I started looking at the aging studies. And now, very recently we did those studies, and 8-month and 12-month mice, they are knockouts are significantly obese, and they have like subcutaneous gonadal fat, and everything is increased. So with that, of course, we also verified the potential transcriptional regulatory role of P311, and P311 binds to master regulator promoter, PPR to promoter. As this morning mentioned, we use the different luciferase assay models and GPSs. And finally, at the end of the day, what we need to know is P311 is affecting both quantity as well as quality, as shown by these genetically modified mice. Right now, I am developing like tissue-specific knockouts, and we are going to like do some more studies and present some additional. So now we are like focusing on the phenotype modulation between these different types of adipocytes regulated by P311, and also we are focusing on like new drug candidates based on like traditional medicine and ethnobotany and all those things. So with that, I'm going to like thank you all, and I thank you, like Chair, for letting me go more than like 3 minutes, I believe, more than, and these are some of my collaborator students, and this is my PhD graduate school team where I started medicinal plants work and diabetes work. With that, I thank you all.
Thank you, Dr. Badri, for discussing your wonderful work, and also good luck for your future plan. So there is time for one question, if there is any burning question.
So what is the molecular weight of this P311?
It's a 69 amino acids, 8 kilodaltons.
So what was the reason you checked this protein only by ICC and ISC?
Oh, actually, we have Western blots and all that. I was like cutting down a lot of slides. So,
Okay. Thank you so much.